Today we'll be talking about this paper from ACS Pharmacology and Translational Sciences titled Investigation of the Structure-Activity Relationships of Psilocybin Analogues. And being kind of a pharmacologist with a lot of background in chemistry, I really just like structure-activity relationship papers. Hopefully I don't bore you guys with all the structure-activity stuff I talk about on this channel, but let's get into it. The first experiment the researchers ran in this paper was where they basically took 4-HO-DMT or psilocin derivatives, injected them into mice, and looked at the head twitch response exhibited by the animal. I've talked about in other videos the idea that if you inject a psychedelic drug into mice, they will twitch their head. The more times they twitch their head in a given time period, the more potent the drug is, which is pretty interesting. So the EC50 value is this number. You don't really have to focus too much on the values. What you need to know is that an EC50 value that's closer to 0 means it's more potent. An EC50 value being further away from 0 is less potent. If we take one of the methyl groups from 4-HO-DMT and change it to an ethyl, the potency slightly decreases. If we change that methyl to a propyl group, as in 4-HO-MPT, the potency also decreases by quite a big factor here. And then if we change the methyl to a isopropyl, once again the potency decreases, basically suggesting that if we take one of those methyls and make that side chain larger and larger and larger, we see a loss of potency, likely due to a change in the binding site when you add these derivatives inside the serotonin to a receptor. If we then take the two methyl groups, change them to two ethyl groups, the potency decreases by about a factor of 2 by the EC50. If we change one of those ethyl groups to a propyl group, the potency stays the same. I thought it would increase just a little bit, due to the fact that 4-HO-DMT and 4-HO-MET show different EC50 values when we change the methyl to an ethyl group. So this surprised me. When we take both of the ethyl groups and change them to propyl groups, as in 4-HO-DPT, the potency increases by a pretty big factor. This makes sense because we've seen this sort of trend where if we make the side chain bigger on the dimethyl groups, the potency decreases. And then with 4-HO-DIPT, we're just changing the propyl groups to isopropyl groups. And we can see that the potency also decreases, indicated by the increase in EC50 value. The last drug they measured was 4-HO-MALT. This is basically if you take one of the methyl groups, change it to an ethyl, and then put a terminal alkyne on there. This is this compound 4-HO malt, has an EC50 value of 0.91 milligrams per kilogram. I was kind of surprised by this because I would have thought that 4-HO-MALT and 4-HO-MPT would have had very similar EC50 values, but I guess it turns out when you take the propyl, as in 4-HO-MPT, and change the terminal alkane to an alkene, the potency decreases. That kind of surprised me. So case in point is basically that when you take the side chain that has the dimethyl groups and make them bigger, the potency of the drug decreases, which probably makes sense because this would change how it fits inside the serotonin receptor. In the next set of experiments, the researchers looked at the 4-acetoxy psilocin derivatives in terms of the head twitch response. Basically the same experiments, different substitution. Instead of having the alcohol at the 4 position, as in psilocin, we now have ester at the 4 position, making it 4-ACO-DMT. This is quite an interesting compound, and a lot of people seem to believe that or subscribe to the idea that 4-ACO-DMT inside the body undergoes enzymatic change from the ester depicted here to the alcohol, as in psilocin. But it is known that 4-ACO-DMT by itself does have psychedelic effects. So it doesn't necessarily have to undergo hydrolysis for this drug to have psychedelic effects, but likely it does. So we've got the EC50 values listed. If we change the methyl to an ethyl, the potency decreases, same as before. If we change the methyl to a propyl, the potency decreases, same as before. If we change the propyl to an isopropyl, the potency also decreases, same as before. If we change the dimethyl to a diethyl, as in 4-ACO-DET, the potency would decrease going from a 4-ACO-DMT to 4-ACO-DET. And then this is interesting because if we go from the ethyl to the propyl, the potency actually increases, indicated by a decrease in EC50, which would be an increase in potency. That's actually flip-flop from what we saw in the previous analogs. With 4-ACO-DPT, we change both the groups to propyl groups, and the potency from 4-ACO-DET to 4-ACO-DPT actually, quite interestingly, increases, which is different from before, indicating that if we go from a diethyl to a dipropyl, for whatever reason, the potency increases when you have the ester functional group in the 4 position. Not exactly sure why. Definitely something interesting going on there. And the last compound, 4-ACO-DIPT, which we change them to isopropyl groups, the potency decreases by quite a big margin here. So the general idea is quite interesting. When we have the ester at the 4 position, the compounds behave slightly differently than if we have the alcohol at the 4 position. And lastly, the authors of this paper looked at basically agonism at the serotonin subtype receptors with all of the various psilocin derivatives, specifically at the serotonin 2A, 2B, and 2C receptor. If we first look at serotonin, which is 5-HT, we see the EC50 value listed for 2A is 0.26 nanomolar, at the 2B receptor at 0.21 nanomolar, and at the 2C receptor at 0.25 nanomolar. So we can basically compare each drug's effects at the various subtype receptors to say which one displays the highest potency and which one displays the lowest potency. I've placed a blue box around each drug at the receptor with the highest potency, and a red box around the receptor with the lowest potency. And what we can generally gather is that the highest potency for all of these compounds is shown at the serotonin 2B receptor, and the lowest potency is shown at the serotonin 2C receptor. Furthermore, we can see that if we compare the serotonin 2A and 2B receptor, they're pretty similar in potency. But if we compare the 2A, the 2B to the 2C has a massive jump off in terms of a decrease in potency. And lastly, we can see that if we compare the ester derivatives, such as 4-ACO-DMT to psilocin, we can see that the potency from psilocin, let's say for example the 2A, goes from 2.40, and if we go to 4-ACO-DMT, it's 103. So we can really speak to the idea that this ester substitution does drastically reduce the potency of the drug at all of the serotonin subtype receptors, which is quite interesting. Finally, let's go over just a quick summary of what we learned from this publication. We learned that when the dimethyl groups in psilocin are changed to larger alkyl groups, the potency decreases in the head twitch response, which is an animal model. Most of the psilocin derivatives measured at various serotonin receptor subtypes show the greatest potency at the serotonin 2B receptor and the lowest potency at the serotonin 2C receptor. We saw that the ester substitution at the 4 position shows a noticeable decrease in potency compared to the alcohol substitution. For example, psilocin has a much greater potency than 4-ACO-DMT.